Hi everyone, my name is Meghan.Donnelly and I am the Nutrition Services Manager and Registered Dietitian at Dr. Shar USA. I hope you're all staying healthy during this time. Today I'm happy to share with you what I hope is an informative presentation on digestion and gut health in celiac disease. Here is my contact information. Feel free to email me with questions after the presentation. I also quickly want to address a few logistical items before we get started. First, your certificates of participation, along with a copy of the slides, will be emailed to you after the webinar has ended. The webinar has been approved for one continuing education unit by the Committee on Dietetic Registration. If you do not receive a certificate via email, please email me. We'll also be recording the webinar and posting it on the Dr. Shar Institute website for on-demand viewing. All of the DSI information will be provided in the email following this webinar. Feel free to type your questions throughout the presentation in the Q&A tab, and I'll try to address them as time permits at the end. So here are some learning objectives for the presentation. After this module, you should be able to define celiac disease, its causes and genetic factors, explain the mechanisms of celiac disease pathogenesis, discuss the key drivers of dysbiosis that may be a factor in celiac disease susceptibility, and describe celiac disease susceptibility related to bacterial imbalance. All right, so here's our outline. This is what we're doing today. To get started, let's briefly discuss some of the basics of celiac disease, though I'm sure a lot of you are fully aware about this. Celiac disease is an immune-based reaction to dietary gluten causing injury to the small intestine. It affects those with genetic predisposition and resolves with exclusion of gluten from the diet. The prevalence of celiac disease is 1 to 2% in Western countries, but the diverse clinical picture of the disease makes diagnosis challenging for physicians, and it can take anywhere from a few months to several years to diagnose. A delay in celiac disease diagnosis is associated with decreased quality of life and an increased number of doctor visits, sick days, and use of pharmaceutical agents. Individuals that have a first-degree relative with celiac disease, type 1 diabetes, or other autoimmune thyroid diseases have an increased risk of celiac disease. Those who are newly diagnosed should inform their first-degree family members of the potential increased risk of developing celiac disease and recommend screening. Those diagnosed with type 1 diabetes or autoimmune thyroid disease should also be screened. Non-responsive celiac disease is defined as continuing to have persistent symptoms, elevated antibodies, or small intestinal damage, even after following a strict gluten-free diet for 6 to 12 months. Studies of adults with non-responsive celiac disease suggest that 36 to 82% of cases may be related to the gluten-free diet, non-compliance, or inadvertent gluten ingestion. Persistent gluten-free diet may be caused by other undetected disorders and complications. Celiac disease causes damage to the small intestine in the form of villous atrophy. This means that the villi in the small intestine are flattened and the epithelial cells are damaged. This leads to reduced nutrient absorption and a variety of symptoms. The normal intestine has elongated villi, which is critical for digestion and nutrient absorption. So in order to understand celiac disease pathogenesis, it's important that we have an understanding of what is actually involved in the gluten digestion process. As dietitians, we all know that food digestion really involves 4 major parts of the body: the mouth, the stomach, the small intestine, and the large intestine. I'm going to be going into detail about gluten digestion at each step. It's important to remember that gluten is a protein, and therefore the main sites of gluten digestion include stomach and small intestine. Large protein strands are broken down into smaller strands and eventually amino acids. So digestion begins with mastication of food in the mouth. In the human digestive system, food enters the mouth, and the mechanical digestion of food starts by the action of chewing and the wetting contact of saliva. Saliva is a liquid secreted by the salivary glands. It contains salivary amylase, an enzyme which starts the digestion of starch in food. After undergoing mastication and starch digestion, the food will be in the form of small, round, slurry mass called bolus. It will then travel down the esophagus and get into the stomach by the action of peristalsis. Gastric juices in the stomach starts the process of digestion. These juices mainly contain proteases, hydrochloric acid, and pepsin. These are responsible for breaking down longer protein chains into smaller peptides. Because the gluten protein has a high proline and glutamine content, it is somewhat resistant to this protein digestion in the stomach. This is related to the absence of bacteria containing propyl endopeptidases. So after about 1 to 2 hours, the stomach produces a thick liquid called chyme, which enters the duodenum and mixes with digestive enzymes from the pancreas and bile juices from the liver, eventually passing through the small intestine. Now we know that the small intestine is the major site for digestion and absorption of nutrients. It's more than 20 feet long in adults and consists of 3 parts, which are the duodenum, jejunum, and ileum. Digestion takes place in the lumen of the small intestine, and absorption takes place at the small intestinal wall. The lining of the small intestinal wall, called the mucosa, contains the villi, which are those finger-like projections that absorb nutrients and increase surface area of the small intestine. That's what we were discussing before. Each villi has a border of microvilli, which, as I mentioned, facilitate absorption and contain enzymes that digest specific food components. This barrier also prevents bacteria, parasites, and other toxins from entering the body. So here's a short overview of the structural components of the intestinal epithelial barrier. I'm just going to quickly define a few key terms here so that we're all on the same page for the next few slides. I know most of us haven't taken cell biology or anatomy in a while, so I hope this is helpful. So enterocytes facilitate absorption of nutrients and protect the epithelial surface by secretion of antimicrobial proteins. Goblet cells are mucus-producing cells. Pannus cells protect the host from bacteria and regulate epithelial renewal. The lamina propria is the supportive layer of conjunctive tissue and lies underneath the epithelium. This provides defense against harmful substances. An even closer look at the structure of the epithelial junctions is shown on this slide. The epithelial barrier is held together primarily by tight junctions and adherens junctions. Tight junctions are located at the apical surface of the enterocytes, and they act as a gate in the paracellular transport of ions, solutes, water, and cells. Tight junctions are highly dynamic structures whose degree of sealing varies in response to external stimuli, as well as physiological and pathological conditions. The tight junctions are composed of transmembrane proteins, occludins, which are involved in tight junction maintenance and assembly, and claudins, which are the sealing proteins involved in permeability. The function of adherens junctions is just what it sounds like. They're responsible for cell-to-cell adhesion. All right, so just to remind everyone where we are in the body at this point, the small intestine is the main site of absorption. And it's important to note that all of the processes I just described occur at the epithelial barrier of the villi. Since the villi are flattened in celiac disease, obviously the normal digestive process is extremely altered in these patients. Okay, moving on to the large intestine. The large intestine also helps in absorption of vitamins made by bacteria that normally live in the large intestine. These are friendly bacteria called commensal bacteria. There are over 700 species of bacteria that perform a variety of functions in the body. These commensal bacteria in the large intestine break down the undigested polysaccharides or fibers in the diet into short-chain fatty acids. These can be absorbed by the large intestine by passive diffusion. The bacteria also produce gas, which is primarily a mixture of nitrogen and carbon dioxide. These bacteria also produce large amounts of vitamins, most important of which are vitamin K and biotin. When vitamin intake is low in the diet, this can be an important source of these vitamins. So a person who depends on absorption of vitamins formed by bacteria in the large intestine may become deficient if treated with drugs like antibiotics that kill the commensal bacteria. All right, so now that we've gotten through this little anatomy lesson, I want to specifically talk about celiac disease pathogenesis and its impact on digestion. The pathogenesis of celiac disease is split up into a series of 3 events. These are the luminal and early mucosal events, the activation of pathogenic CD4 T cells, and the subsequent events leading to tissue damage. So we're going to go through each one of them separately. First, as a reminder, celiac disease is caused by an immune reaction to gluten, which damages the small intestine. Gluten is made of 2 proteins, gliadin and glutenin. The very high glutamine and proline content in the gliadins and glutenins renders these proteins relatively resistant to proteolytic digestion by gastric, pancreatic, and brush border enzymes in the intestine. Due to their size, they are not readily absorbed and accumulate in the gut lumen to cross the epithelium. The image on this slide takes us back to that small intestinal epithelial barrier. You'll see on the left side, in the gut lumen, the large undigested gliadin peptides cross into the small intestinal mucosa and are deaminated by tissue transglutamase. Once they cross, these altered peptides in the TTG activate CD4 T cells, generating antibodies against gliadin and TTG. Under normal conditions, proteins are mostly hydrolyzed by gastric and pancreatic peptidases in the GI tract, resulting in smaller peptides or amino acids. Nonetheless, the relatively poor digestion of these proteins is not alone sufficient to cause celiac disease. There is no known difference between healthy individuals and those susceptible to developing celiac disease in their ability to digest these proteins. So, healthy people and people with celiac disease all have trouble breaking down gluten. The failure to degrade these proteins may be exaggerated in the small intestine of individuals with active celiac disease who have epithelial cell brush border injury. Propyl endopeptidases produced by bacteria can digest these proline-rich gluten peptides. Patients with celiac disease may be deficient in these enzymes. The second stage of celiac disease pathogenesis is the activation of CD4 T cells. This looks really complicated, but I'm hoping to simplify it here for you. Basically, the gliadin peptides are given a negative charge by TTG. From here, the gliadin peptides bind to antigen-presenting cells, otherwise known as APCs, which triggers the T cell response and immune cascade, and eventually leads to intestinal alterations, including villus atrophy, crypt hyperplasia, and increased epithelial lymphocytes, all of which we typically see in classic celiac disease. Now, we all know that there's a genetic component of celiac disease. HLA, DQ2, and 8 are genes on chromosome 6. Despite the fact that 30% of the population carries the gene, only 1% actually develops active celiac disease. So it is important to note that the HLA, DQ alleles are necessary, but not sufficient for celiac disease diagnosis. In the digestive process, HLA-DQ serves as the cell surface receptor on APCs. The deaminated gliadin peptides that have just crossed the epithelial barrier strongly bind to HLA-DQ2 and 8 genes, which enhance gluten-specific T cell response in people with celiac disease. In other words, when antigens are detected, like gluten, the antigens attach to the HLA-DQ receptors and cause an immune response. All right, so now we're going to talk about the role of gut microbiota in celiac disease development. This is really an exploratory area, so there's still a lot to learn here, but much of the research is devoted to this topic, so I thought I would cover it. The human gut microbiota is comprised of trillions of microorganisms that influence our health and well-being. It's sterile at birth, but colonization begins almost immediately, through the mother's vaginal and fecal microbiota, as well as the surrounding environment. The colonization of diverse microbes continues with breastfeeding, diet, and the environment the baby lives in. Further colonization of microbes is due to genetics, lifestyle, geographic origin, age, eating habits, and microbial exposure such as antibiotics and probiotics. For our purposes, microbial dysbiosis refers to the imbalance of beneficial and pathogenic bacteria in the gut. Changes in the gut microbiota may be related to mode of delivery at birth, diet changes, infection, medication use, and many other things. These microbiota play a critical role in gut health and immune health. There are a lot of questions surrounding microbial dysbiosis and celiac disease, and there's some debate about whether celiac disease is the result of microbial dysbiosis or the cause of it. Several factors in gut microbiota development may affect your risk of developing celiac disease. For example, mode of delivery may be a factor. Babies delivered via vaginal birth are exposed to more bacteria and have a more robust gut microbiome. Thus, there may be an increased risk of celiac disease in C-section newborns, but not all studies support this. Additionally, there are some retrospective studies that show longer duration of breastfeeding and maternal gluten intake may reduce or delay celiac disease onset. However, this is definitely a controversial topic and is likely not conclusive. One study by DePalma studied 164 newborns with one first-degree relative with celiac disease. They looked at HLA genotype and modality of feeding, so breastfeeding or formula. They found a different gut colonization according to type of feeding, so lower beneficial bacteria and higher harmful bacteria in the formula-fed infants. It's important to remember that breast milk contains immunoprotective compounds, so without these, a genetically predisposed child may in fact be at higher risk for celiac disease, although this is definitely not confirmed and still remains an area to be researched. Other studies show that breastfed babies carrying a high genetic risk, HLA-DQ2, had higher amounts of bad gut bacteria compared to those with lower genetic risk. This suggests that host genotype may modulate the gut microbiota and increase disease risk. Studies are currently being done to study the role of the gut microbiota as a contributor to the risk of celiac disease onset. There is an Italian study that reported that intestinal infections were strongly associated with the onset of disease and were further strongly associated with antibiotic use. Moreover, early age infections and infants' antibiotic intake is also reported as the cause of dysbiosis and alterations in lymphocyte subpopulations, which can be correlated to disease activity, such as increased intraepithelial lymphocytes and villus atrophy. Such antibiotic-induced dysbiosis was characterized by decreased counts of Bifidobacteria longum and increased counts of bacteriocetes. And these are basically, decreased good bacteria and increased bad bacteria. In summary, these are all ways in which environmental gut microbiota alterations may lead to increased risk of developing celiac disease. All right, so now we're going to talk about how the gut microbiota affects celiac disease pathogenesis. So we're going to look at gluten digestion itself, intestinal permeability, and host immunity. Just a few reminders. Gluten proteins are not degraded well by peptidases in the GI tract, so the large gliadin peptides enter the small intestine relatively unaltered. These peptides contain immunogenic sequences that induce immune response. This model depicts how imbalances between pathobionts and coarse commensals in the proximal small intestine can affect susceptibility to celiac disease in a genetically predisposed host. For example, partially degraded gluten peptides on the left side here may be metabolized by pathogenic bacteria such as P. aeruginosa. This bacteria digests the peptides into shorter amino acids. Peptides longer than 9 amino acids strongly stimulate gluten-specific responses in celiac disease-relevant T cells isolated from genetically positive celiac disease patients, which actually subsequently increases the immunogenicity of the 33 polymer peptide in celiac disease patients. On the other hand, studies show that P. aeruginosa modified gluten peptides can be further detoxified by beneficial bacteria, such as the lactobacillus species. The metabolic activity of lactobacillus may mitigate this immune response and be beneficial, and you can see this on the right side here. By incubating partially digested gluten, Lactobacilli strains decreases its immunogenicity to gluten-specific T cells from celiac disease patients. One study showed that the immunogenic peptides produced by P. aeruginosa proteases are further broken down and said to be less immunogenic in the presence of lactobacillus. This mechanism may partially explain the relationship between microbial dysbiosis and susceptibility to celiac disease in a genetically predisposed host. I'm just reminding you this is just a proposed mechanism, so definitely more research needs to be done here, but interesting food for thought. All right, moving back to the small intestinal epithelial barrier. So zonulin is a protein that modulates the permeability of tight junctions and enterocytes, and it's overexpressed in celiac disease patients. Higher levels of zonulin are associated with reduced tight junction integrity. Gliadin peptides bind to the CXCR3 receptor on the intestinal epithelium, which stimulates zonulin release and leads to reduced tight junction integrity. Then, gluten peptides pass through the intestinal barrier, causing an inflammatory response. Interestingly, the receptors that gliadin molecules bind to may be affected by dysbiosis of the gut microbiota. For example, one study showed that a CXCL10, a ligand for CXCR3, is overexpressed in the small intestine of celiac disease patients. The CXCL10 CXCR3 axis has been shown to be activated by pathogens, suggesting that this mechanism may play a role in initiating gluten-induced inflammatory processes in the small intestine. But why is this even a problem? Why is it a problem when the tight junctions break down? Tight junctions impede the entry of pathogens and toxins from crossing the small intestinal barrier. So sustained inflammation and infection can cause deregulation in the expression of the tight junction molecules, which allows toxic and immunogenic substances into the lamina propria. All right, and moving on to the host immunity. All right, so this image shows the connection between gluten microbes and the intestinal immune regulation. The first column over here shows a healthy microbial environment. There's a balance between beneficial bacteria, these green ones, and harmful bacteria, which are the gray ones. The beneficial bacteria prevent the harmful bacteria from adhering to the intestinal mucosa, causing and preventing damage to the intestine. The middle column demonstrates how harmful environmental infectious agents, which are shown in red, battle with the beneficial bacteria to adhere to the intestinal mucosa. If harmful infectious agents bind, they have the potential to disrupt the intestinal barrier and its functions, such as the tight junctions. They do this by activating different inflammatory pathways at the intestinal mucosal surface. This opens the door, literally, for the intestinal barrier to be exposed to the dietary antigens, such as gluten. Increased permeability allows dietary antigens to travel past the intestinal barrier and encounter the immune cells. Thus, infections may set the stage for active celiac disease onset. The gluten peptides, which are shown in blue, bind to HLA-DQ and generates an immune cascade that eventually leads to tissue damage and gluten intolerance. The last column shows how antibiotics given to patients to treat infections has a role in dysbiosis. Antibiotics eradicate infections, shown in red, as well as beneficial intestinal microbes, shown in green, leading to dysbiosis. In this situation, the opportunistic harmful microbes, shown in gray, may prove to be dangerous because beneficial microbes can no longer protect intestinal mucosa. This results in harmful microbes adhering to the intestinal epithelial cells and creating disease-susceptible microenvironment. All right, I'm not going to spend a lot of time on this slide, but I wanted to put it here to show the other ways that microbial dysbiosis affects mucosal immunity. Basically, any form of dysbiosis can generate an immune response, which can actually cause structural changes in the host epithelial cells. Of note, CD14 tends to be increased in celiac disease patients, suggesting that there's some inflammatory response going on here. All right, changing gears a little bit. We know that diet changes affect the gut microbiota as well, so not surprisingly, so does the gluten-free diet. In one study of healthy individuals, there weren't really any differences in the gut microbiota composition with gluten elimination alone. Here it really was that, even though if the overall diet stayed relatively the same and they just eliminated gluten, there wasn't too much of a change. The researchers concluded that the alterations may be related to reduction in polysaccharide intake, such as fructans, which are prebiotic. Additionally, studies show that the gluten-free diet may only be partially effective at restoring the gut microbiota in celiac disease patients. Many studies show that there are differences in the abundance and diversity of lactobacillus and bifidobacterium in both treated and untreated celiac disease. These are typically affected by carbohydrate and starch metabolism. However, there are significant limitations with many of these studies, including small sample sizes with clinical variability and presentation of celiac disease. Microbiome assessment is also a challenge. For example, the microbiome assessments in many of these studies were directed at specific strains and not the entire microbiome. This is a problem because you don't really know what's being affected and what's driving these changes. Finally, you always have to take into account that individual diets vary greatly, which is really difficult to control for. Everyone's individual microbiome is going to be very different depending on their environment, and we can't really do studies where we put everyone on the same diet, so it can be really a challenge. All right, so now we're going to talk briefly about some of the potential therapeutic targets in celiac disease as they relate to gut health. So there are a number of therapies in development aimed at the epithelial barrier, which makes sense given all the information we just reviewed. I'm certainly not an expert on this topic, but I do want to mention a couple of them here. One is larazotide acetate, which essentially inhibits zonulin and reduces tight junction alterations related to gliadin exposure. This is definitely still in the early phase of research, and we need to do more, but really interesting therapeutic option. I do want to spend more time discussing the role of probiotics, since I think a lot of us are dietitians here and most likely get a lot of questions about probiotics. As a reminder, these are live microorganisms that confer a health benefit on the host. They improve the microbial profile of the host and produce substances that block and degrade pathogens, compete for nutrients, and regulate immunity. Some in vitro studies show that the lactobacillus species reduce gluten concentration and immunotoxicity when added to sourdough. In human studies, celiac disease patients were challenged for 60 days with lactobacilli and pre-digested gluten. These patients did not experience worsening of symptoms or changes to the small intestine, suggesting that this type of therapy is a possible potential adjunct to the gluten-free diet. I want to highlight here, this does not mean that people should consume gluten-containing foods if they're taking probiotics, but it's just an interesting area to study. Oops, sorry, went ahead. There are some general benefits to taking probiotics. They may stabilize tight junctions and restore gut microbiota in conjunction with a gluten-free diet in newly diagnosed celiac disease patients. So we know that, a lot of times people, when their intestines are healing, they need to replenish the good gut bacteria. So probiotics may be a helpful tool to do that, along with a well-balanced diet that's high in fiber. I do want to point out some significant limitations in celiac disease probiotic studies. The first is that the microbial picture of each celiac disease patient and any individual is very different depending on environmental factors. It is possible that probiotics help control inflammation and symptoms, but probiotics are largely unregulated and contain varying amounts of relevant species. Additionally, it's unclear if specific strains or multi-strain probiotics have any benefit, so we don't know if it's one type of bacteria that's causing a benefit or if you're better off with a broad spectrum. It's really, really unclear, and I think it's definitely something that needs to be studied more. All right, phew, finally made it. So in summary, the gut microbiota is highly individualized in its colonization and response to the environment. Gut microbiota play a role in gluten digestion, intestinal permeability, and host immunity in individuals with celiac disease. Gut dysbiosis may contribute to the pathogenesis of celiac disease, and potential therapies may be targeted at the gut microbiota structure and function. All right, so now we're going to go through some of the self-assessment questions. So just type your answers into the Q&A box as we go, and I'll try to get a general consensus before giving the answer. All right, so celiac disease is strongly associated with which one of these? All right, most of you got that one. The correct answer is A. So celiac disease is strongly associated with the HLA, DQ, and 8 molecules. The pathogenesis we know is related to genetic factors. All right. TG2, trissugarin transglutamase, acts on which parts of gluten? All right, so a few mixed answers here, but it looks like, yep, it looks like most of you are getting this one right. The correct answer is B. So TG2 converts neutral glutamine to negatively charged glutamic acid. It's released into the intestinal mucosa and causes enzymatic modification of the gliadin peptides. TG2 deaminates the peptides and causes TG2-induced enzymatic change that unmasks the most immunogenic epitopes of gliadin. By increasing the expression of negative charges, this increases the affinity to bind to the genetic markers HLA, DQ2, and 8. All right, gluten peptides cause the release of what protein? And what does it do? All right, some mixed answers, but for the most part, I'm seeing the correct one. The correct answer is C. So zonulin rearranges the cytoskeleton of the cell and modifies tight junctions, leading to increased intestinal permeability. Okay, last one. What are the key players of dysbiosis that may be a reason for disease susceptibility? All right, good. Most of you got this one. So the correct answer is B. The key players of dysbiosis are antibiotics and infections. Early age infections and infants' antibiotic intake has been shown to be a cause of dysbiosis and alterations in lymphocyte subpopulations that can be associated to disease activity. All right, so now I'm going to open it up to questions. So again, type your questions into the chat box and I'll try to get to as many as I can. So I have one here about, is there a way to delay the onset of celiac disease in genetically susceptible populations? So most of the research in this area is really, it's a little bit unclear. Some studies show that there is some benefit to delaying gluten ingestion in these populations, but for the most part, I think it's really important to consider that you shouldn't be restricting gluten unless you have to. So for the most part, no, the research does not really suggest that you need to restrict gluten if you're genetically susceptible. And in fact, I think it's better to follow a well-balanced diet, which includes gluten, before there's diagnosis. Also keep in mind, genetic screening is not necessarily done unless you have a first-degree relative with the disease. So I'd say encourage patients to follow a balanced diet and not worry about it until they're experiencing symptoms or increase antibodies in the blood. Okay. So someone's asking about diagnosis in children and what is the process. So diagnosis is really the same. The diagnostic process is kind of the same for all populations, including children. So it'll start with a blood test. And if your antibodies are elevated, then the gastroenterologist will perform a biopsy of the small intestine. They need the elevated antibodies and the biopsy to confirm the diagnosis. Okay, someone's asking about symptoms. Oh, and if a symptom can be a skin rash only. So yeah, people can present with a variety of symptoms. The most common ones, especially in children, are really gastrointestinal related, but some others might be poor growth and a skin rash called dermatitis herpetiformis, which is what this question is getting at. So yes, a child may present with just the skin rash and no other symptoms. That's very possible. But a lot of times you have to be looking at other things, nutrient deficiencies, poor growth, and gastrointestinal symptoms. Those are all really common in children. All right. How do you select a probiotic with lack of regulations in probiotics? So this is a really good and complicated question. There's a lot of different probiotics out there, and I think the most important thing is if you're recommending one, is to recommend a multi-strain probiotic from a reputable company. There are some private companies that certify probiotics for contents, and I think it's important to make sure that you're getting one from a company that does testing and really pays attention to the quality. The way you know that is really just looking at their individual websites. So if there's a brand that you see that you think is reputable, stick with that one, and typically you want to recommend a multi-strain probiotic. All right, and we have a question about genetic markers. Can you have celiac disease without the genetic marker? You need the genetic marker in order to have celiac disease. So if you have maybe a questionable biopsy and serology, then it's possible, but you have a negative genetic marker, then really it's possible that you have something called non-celiac gluten sensitivity. These are required genes. It is a genetic condition. So again, if you have a questionable serology and biopsy, a lot of times they'll do the genetic test to rule celiac disease in or out, but this is really not always done. All right. I am not seeing any more questions. I'll just wait a couple more seconds. Okay. All right, so feel free to email me if you have additional questions. I'm happy to do my best to answer all of your questions. So if something I missed or something you want more detail on, feel free to email me. Here's my references. I do just want to touch briefly on what we do here at Dr. Shar. We are a dedicated gluten-free company committed to providing high-quality gluten-free food for over 30 years. We are always focused on nutrition and health, and so we try to use as many whole grains whenever possible, and sourdough really, and high fiber. Most or all of our breads are good or excellent sources of fiber, and so this is really all aimed at providing the best quality food and also promoting a healthy gut. So we try our best here at Shar, and here's some more information if you're interested, or you can always check out our website. All right, and just a reminder, I will be emailing you all a copy of the slides and sending out your certificates of participation. You can also view this webinar later on the Dr. Shar Institute website, which I will provide the link to. So thank you for listening, and I hope you enjoyed the presentation.